Our next session is Translational Research, the path from evidence to intervention. The moderator is Dr. Mark McClellan, the Robert J. Margolis Professor of Business, Medicine, and Policy, and Director of the Duke-Margolis Center for Health Policy at Duke University, and a member of the Research America Board of Directors. Mark will be joined in conversation with Dr. David Reese, Executive Vice President for Research and Development at Amgen, to discuss what defines translational research. Mark?
David, it's good to be with you today to talk about translational research. Now, there are a lot of terms that come up for the many different phases of product development, from discovery all the way through effective patient access. In your career in the academic setting and the nonprofit setting, and now in your current position as Executive Vice President for R&D at Amgen, you focused on translational research. Can you explain to all of us how you think about translational research and its critical role in the development process?
Great. And it's great to talk to you today, Mark. A critical topic here. I think there are probably as many definitions of translational research as there are translational researchers in one way, but I like to think about it as translating the translational part from the laboratory to the clinic in our domain. So what does it take to get an idea from the laboratory? In the case of drug development, how do you start a molecule on the way to becoming a medicine? What's required to move that into the clinic? And then, in the case of diagnostics, for example, how do you go from an idea, an assay, on the road to the introduction of an actual diagnostic? So it's that fluid interface between the laboratory or preclinical work and clinical work, and it should be bidirectional.
And in terms of participants in this stage of the development process, so seems like an area where there is a lot of public involvement. We have a national institute that focuses on translational research, but also involvement of academic organizations, companies that are at the early stages, as you said, of turning these ideas into products. How does that interface work, and has that been evolving over time?
I think it is evolving over time. That that interface is now an ecosystem, the most successful ecosystem in the world, in the United States. One way to think about it is that the public sector is involved primarily, not exclusively, but primarily in generating the basic science that forms the foundation for what will come, whether that's drug development, new therapeutic approaches, diagnostics, all the sorts of things that we've been talking about. Industry is focused really on applied science in many ways. Again, how do you get from an idea to an actual medicine? There are a lot of steps in between. Some are relatively specific to industry, some are shared across that ecosystem. Academia dwells along this entire spectrum from, of course, conducting basic science in the crown jewels of the academic laboratories across the country, to doing the early phase and later phase work, often in collaboration with industry, that ultimately leads, in the case of drug development, to a drug approval. So it is a complex ecosystem. It is one where all of those elements are required. So you can't chop out one and expect it to work, certainly the way it has worked in terms of the progress that we've had against human disease.
And do you see that evolving? I can see the critical role for each, the federal funding and research support for the basic science and for scientific advancements that help with translation across multiple products. Certainly lots of active academic involvement, as well as companies. Does it look the same today as it did a few years ago? What kind of trajectory is this field on?
I think it is evolving in that the boundaries are getting a little more fluid themselves between these domains. And of course, many new companies now are spin-outs from the academic environment. And they are then designed to take an idea forward and do the things that typically can't be done in a university, or to be honest, shouldn't be done in a university because it's simply not efficient, and there are better ways to get those things done. So I think those boundaries are blurring, but you can still see recognizable chunks of this ecosystem.
Mm-hmm. And over the years, people have talked about the gap, the chasm, this valley of death between good research ideas in the lab and getting that human testing and clinical development going. Is that evolving at all too, or is that still a big challenge to fill that gap and lots of development difficulties along the way?
It is evolving. I think it's evolving spurred by developments in technology, but it is still in many ways the most formidable obstacle, and that valley of death does still exist. There are certain challenges. For example, when you're going into the clinic, if you've got a novel therapeutic or a disease where there has not been much development, for example, what do you use as endpoints? How do you know that you're achieving in the clinic what you want to achieve with that molecule? It may be just a very hard question to answer. Without answering that, though, how do you then layer in the next level of investment, which is often, an order of magnitude greater than what it takes to do those first experiments? One of the things that we often do, and of course we collaborate with many, many academic research groups and small biotechs, is say, okay, what's the killer experiment? What's that next set of data that you need to believe that you're actually achieving what you set out to do? To me, that's the heart of translational research, and always coming back to that simple question. What's the killer experiment? What's the data package you need to see to think that this actually has some merit?
And are there some new developments in data science, AI, standards or markers that have made that process easier? Again, experiments don't always succeed, but the faster you can get that killer experiment done and get to failure or success, the better.
Yeah, to me, we are actually at a hinge moment where we're going to see a qualitative change in how this is done. Again, that hinge moment is being propelled by technology. We've seen a genetics revolution, genomics, but we're now layering in many other forms of what we call omic technologies, proteomics, metabolomics, transcriptomics, to really create an understanding of the fundamental biology of disease that is greater than we've had before, so that when you get to the clinic, you're loading the dice as much as you can in favor of your approach. It's all a game of probabilities. If you can simply double the probability of a molecule succeeding in the clinic from 10% to 20%, the implications of that socioeconomically, in terms of human disease and public health, are just staggering. And I think we're right on the precipice of being able to do that. And it's everything from artificial intelligence to help us understand protein-protein interactions through this tsunami of real-world data and real-world evidence. We like to call it human data at Amgen, the era of human data. And I think the entities that will be most successful are those that figure out how to ingest, aggregate, and then most crucially, analyze these data to, again, achieve a depth of understanding of human disease that just hasn't been possible before. And we're right on the lip of that. I really think that in my bone marrow.
Well, I want to come back, if we have time, to what that's going to look like. How do we know when we've successfully made that hinge move? But to get there, your emphasis on ingesting, aggregating, analyzing data, doing it in a way that's valid, I assume reproducible, transparent, for acceptance in the research and the medical and the clinical and the regulatory community. That seems like, David, it's going to require more, not less, collaboration across the different groups that we've talked about playing a critical role here: federal funders, academic groups, and industry. And these are all groups that, especially academics and industry, they sometimes have challenges in doing that. Are you seeing the mechanisms of interaction here and the mechanisms of development science keeping up with the science, the scientific opportunities around ingesting, aggregating, analyzing data?
It's a great question. It's one we get frequently. I'm actually quite optimistic about that. I don't think that's the rate limiting step. It's often posited as the rate limiting step, but to me, the bigger rate limiting step is the biology and understanding the basic science. One thing that the pandemic did was it showed us the art of the possible, because you had all of these communities working together in real time, to develop therapeutics and vaccines, of course, in light speed. Well, that actually tells you what's possible. To me, the question is then, how do you try to make that routine? Far more people died of atherosclerotic cardiovascular disease during the last 2 years than died of COVID-19. Where's our sense of urgency? Where are the emergency committees dealing with that? At some point, we need to take these models and translate them themselves into drug development for diseases that have enormous global public health import.
I mean, that's certainly what happened with COVID. And looking ahead, it seems like there may be some opportunities to repeat that, perhaps for atherosclerotic disease, for example. ARPA-H has gotten a lot of attention, aiming to take some new transformational approaches in key areas where there's lots of public health burden on the one hand, but scientific opportunity on the other, which some of these development science areas may relate to. Maybe there's some ways to rethink government-funded research. We just saw Congress pass major bipartisan legislation around platforms for getting the semiconductor industry to a next level of productivity and capabilities. If you had your wish list for policy in this area, what would you like to see to help hasten those kinds of transformative approaches, not just for COVID and a public health emergency, but for all of these high burden health conditions where, as you said, the individual factors, I mean, they really do matter, and they really do present challenges in effective targeted product development?
Yeah, so I think, removing some of these barriers to collaboration, is a must, but these are solvable problems. We know precisely what the problems are. To me, it's then picking perhaps a handful of things, whether it's atherosclerotic cardiovascular disease, cancer, of course, gets a huge amount of attention. But there are, a handful of diseases that account for 60-70% of the public health burden around the globe. How do you create, moonshot type thinking around those disorders, some of which we actually know some of the answers already. It's actually on the implementation side, that we lack, as opposed to actually knowing the medical answer to certain questions. So, to me, if I were emperor for a day, to get to your question, I would say, okay, how do we organize ourselves around this so that we're going to have a huge public health impact? What are the timelines, and what do you need to do? What are, it's a standard analysis. What are the gaps in our knowledge, in our capabilities? How do we fill those in? And, where do we go from here? It's actually, I don't think it's rocket science. I think sometimes we overthink this, but the collaboration and cooperation part actually gives me the least worry.
Mm-hmm. It sounds like it's more the vision and then organizing the support behind the vision, like we showed we could do in the COVID response.
Exactly. I think that's exactly right.
Mm-hmm. Well, just to maybe build on that a bit, you do have this unique career background spanning across all of these sectors that need to collaborate to make development science work, let alone take on these moonshots around taking advantage of the new omics and supporting data for transformational progress. Any insights that you've had as you've worked on these different sides of these same fundamental, challenging issues about how we can work together better to get to some of these goals?
Yeah, I think a lot of, and I have worked on, on all sides of this. I would also say that they're also not as different as we sometimes pretend. The intellectual challenges are often quite similar, and we have the same goal, which is, defeating disease, in one fashion or another. And so, to me, the question is, again, how do you organize this, and how do you actually create things that are fit for purpose? Our most successful collaborations are ones where we have a defined goal that's shared with, for example, an academic group, and then we develop a plan mutually towards that goal. Again, this is not rocket science, but it's surprising how frequently we forget some of these basics. And then, we periodically take stock and we say, where are we in terms of our milestones? Just the way we do with every drug development program internally at Amgen or any company for that matter. I think that's one way to, approach these partnerships, and it tends to make them much more fruitful and satisfying to everyone. And there's also more than enough work to go around. That is not the problem.
That's certainly true. It does seem like, though, for something that's really bold and cross-cutting, could potentially accelerate progress across industry, present new opportunities for research activities, applied research in academic institutions. Government role is very important here. Do you see this as a role that the federal government in the form of NIH or ARPA-H could step up to? Are there other places to look for leadership or collaboration on the federal side?
Yeah, I think there are a variety of models that could be pursued. Certainly, government leadership here, as we saw with COVID, they convened. I sat on one of the committees, for example, that was involved with the active trials. That was multiple branches of government, industry, academia coming together to simply say, what's the best path forward in the epidemic, and how do we quickly get answers? Again, a template for what can be done. Public-private partnerships, I think, can play a huge role, and other bodies such as the Foundation for NIH, FNIH, I think can play a key role. To me, it's figuring out what's fit for purpose for any given challenge.
Yeah, and I hope we can build on those ideas. Certainly, it's a topic of great interest to our colleagues at Research America who have brought us all together here today. Before we finish up, this has been a great discussion about the hinge moment for development science. Maybe we could turn to your related particular area of expertise around cancer research, where, boy, it's been a phenomenal decade and looking ahead, so many opportunities from the omics that you talked about to better understanding of immunologic based treatments and so many other opportunities in the cancer space. Are there any that you're particularly looking at? And I'm not asking you to reveal any Amgen trade secrets, but areas that you're particularly looking at for opportunities for further progress in cancer in the next few years?
Yeah, I think this is also going to break open at another level as well, driven by technology. When you take a slightly longer view than you were articulating, start with the enunciation of the war on cancer about a half century ago. What we've learned in that half century is a pretty good understanding of the molecular underpinnings of cancer. We're now translating that, as it were, into new therapies, and we've seen a variety arise. What we've also seen in the last few years, I think, is a marriage of what you can call targeted therapies. So drugs developed, because of a specific molecular alteration in a tumor cell, targeting that alteration in one fashion or another, and the advent of immunotherapy or immuno-oncology. Now you're going to see the marriage of those two going forward. So I think that's one thing that will propel additional progress. And then finally, to me, crucially, many of those targets that we have identified in the last half century are currently what we call undruggable. meaning our current therapeutic modalities, small molecules, proteins such as monoclonal antibodies or other therapeutics, cannot be used to interdict the pathway or to interfere with the target protein in the right fashion. I think you're going to see a proliferation of new modalities that allow us to drug the undruggable. And that, to me, the undruggable part of the genome right now across all diseases is 80 to 85% of all known targets. So as we develop modalities that can, again, interdict these targets, then things will really blow open. And I think we're going to see that in the next couple decades.
Well, it seems like an area back to where we started, where translational research opportunities to hinge us into a much more productive and much broader range of therapeutic opportunities is coming ahead. David, I want to thank you for the time today to discuss these issues. Any final thoughts before we wrap up?
No, I think it takes a village here at the hinge moment. It's going to actually take probably a city, not a village, going forward. And to me, I'm just extremely optimistic about what the future portends.
Yeah, the potential really is tremendous. David, great to be with you. Thank you very much.
Thanks for having me. Take care.